Well, again, my apology about changing the topic completely and thought of presenting something very different. Well, I'll hope that that I'll be able to keep you awake after this lunch. Yeah. Hi. My name is Imroze. I have just formed my group at Ashoka University around 2 years back, and well, I think those of you who don't know about Ashoka University, it is a university which was founded really recently, and it was started as India's one of the first liberal arts university. And one of the special features that we often take while teaching Ashoka is to contextualize, let's say, a different discipline in some sense of wider philosophical context. And I would say that immunology is something which is not also an exception. Well, I think many of you must be knowing that immunology has been a very philosophical field. Those of you aware of this age-old debate about self and non-self, or biological individuality. Nonetheless, anyway, this is not a philosophy talk. I'll not get into that. So currently I'm working on a field which is popularly known as evolutionary immunology. Well, this is where one can use different evolutionary and ecological concepts to understand immunity and outcome of host pathogen interactions. Well, as I said, it's a recently thing. Well, this according to, let's say, the popular notion, but I would argue against that, because if you particularly take a look at this particular quote, that immunology has been pretty old, and it has had a very deep association with evolutionary biology. And as you can see, the immunology is one of the unique products of Darwinian age, and it was born in the same controversies as evolutionary concepts has gone through. And also, those of you who know this person, Elie Mechnikov, he is known as the founding father of modern immunology. But I think this is also interesting to know that he is actually an embryologist who was trying to understand Darwinian in principles in his experiments. He was trying to understand formation of mesoderm in diverse animal taxa to understand certain design features in animals. And while doing that, he accidentally bumped into a pretty weird molecule which apparently eats up whatever cell debris that he could find in a cell. And he termed this as phagine or something. Well, I think it was known as phagocytes. So phagine in Greek is known as something that eats, and a phagocyte is a molecule which eats up things. And that's how the field started. I think it was known, as I said, is one of the founding father of immunology, and definitely, but originally, definitely he was an embryologist, was trying to follow different Darwinian principles. Also, I think I don't have to tell you that there has been a remarkable progress in the field of immunological understanding in the last 100 years. We now know a great deal of information about cells, molecules, or signaling pathways which could be important for immune responses. But I think there are certain things, or at least this particular question still remains elusive. And why are immune responses shaped the way they are? So practically, I think for the last 100 years, we have been trying to understand immunology from some sort of reaction perspective. It's like A plus B is equal to C, some sort of reaction between host and pathogens, and something happens in the name of immune response. But which is clearly not the case, perhaps, because therefore it becomes more challenging. And I think this question is pretty tough to address at times. And reason being, the astounding amount of diversity that we see in our animals and their response towards infections and disease, right? So, well, I don't think I need to explain, this particular things to this audience. It has been known that immune response vary across species. It also shows tremendous variation within the same species. There has been known evidence of immune response. They keep on varying across individuals within the same species, right? So therefore, the big question, which is somehow not well understood, is why there is so much of variability, or where does it originate from, right? And therefore, and this is something I'm interested in my lab, so which I call an evolutionary immunology lab. So I'm interested in understanding, immune response. As I said, they could be inducible, they could be constitutive, and I try to understand this variation in immune response, to figuring out the cost of such immune responses, right? And this cost could be many. So it could be the fitness cost, it could be more proximate kind of cost. For example, immunopathology, which is the immune cell harm, means the immune response which actually hurts our own cells and organs, right? There could be other proximate damages like DNA damage and mutations. Well, I think immune response causing mutations and one of the causative agent for cancer is quite widely known now. You could also imagine that this cost could also vary under different life history contexts. So it could be vary according to, let's say, the age of an individual, or reproductive strategy of a population, or frequency and nature of the pathogen exposure, or let's say, when a population, they're fighting for resource. And I try to combine this, cost of immunity and also the life history context under which those costs are expressed to understand, the variation, selection, evolution of immune system. So this is my lab is interested in. So I'm not going to talk about all of these things, but I'm trying to tell you a short story about certain topics which are highlighted in bold. For example, aging. I'll try to make you understand that how aging can impact immune response with an evolutionary overtone. And while doing that, I'll also introduce some concept of immunopathology and how immune response can lead to, the DNA damage and mutations, and how it could affect evolutionary processes. So, well, I think this is a question that has been quite popular for many years. I think many people have worked on this, and for the time being, we also know that how immune response could behave with aging. So, I think we all agree that it perhaps deteriorates with aging. However, the question which is most important here is to understand that, why does aging lead to poor immune system? So we all know that aging perhaps contributes to poor immune response, but question is, why does it do it? And therefore, in our lab, we have just started our journey to try to understand this thing as well, and using existing evolutionary theory of aging. So although I think this is also for my personal experience that I have heard people in different talks, they say that aging is quite poorly understood, but I think I would disagree, and possibly you also agree with me that there has been substantial understanding about there has been substantial understanding of the evolutionary theory of aging for the time being. So there has been people working on this topic, trying to understand how natural selection can affect the progression of aging. So one of the predominant hypotheses is that the strength of natural selection, they keeps on declining, with age. And there has been, it has also been modeled that, and this decline in natural selection is also tightly associated with the reproductive schedule. So when the organism is young, so apparently the natural selection is quite strong on it. And when it stops reproducing, the strength of natural selection might be weak. And therefore you could see the regulation on many physiological processes might go really bad with that. So although it doesn't say anything explicitly about immune response, but it would not be so surprising to start with a prediction like this, possibly with aging. So possibly I think in a young individual, immune response could be tightly regulated, right? Because having an immune response which is uncontrolled or inappropriate is going to damage organisms, right? And it could affect organism in many, many ways, especially when we now understand that, immunity is really costly. However, with aging, when the selection is going to be weak on individuals, the immune response control might go weak. As a result of that, you'd see that, all those things were not happening in a young individual, let's say uncontrolled expression of immune response might now happen when the organism is aging. So therefore, I think the first thing that I really want to understand, before getting into more evolutionary question, I really want to understand that what it to be mean by the features of an deregulated aging immune system. So there has been certain criteria that I want to test whether they actually hold true or not. For instance, overexpression, as I was telling you, or sometimes the immune response is cytotoxic, or there could be also increased cost of immunity in terms of immunopathology, right? And also there could be fitness cost. So this is where I started my experiments with so this is a very interesting model system, insect model system. It's called mealworm beetles, Senbera molita, and this is the work that I started in my postdoc. And I was trying to understand how different components of immune response they change with aging. And as you could see that in this graph, with aging, the older individual, it seems that they're being able to, the ability to clear bacterial cells is possibly higher than young individuals, which means they have higher bacterial clearance ability. We kept on moving to multiple other components of immunity as well to understand whether this actually is a true representation or not. We also, try to measure the antimicrobial activity of the hemolymph from the insect. So we extracted hemolymph from young individuals and older individuals and compared their antimicrobial activity. And you could see for the older individual, the antimicrobial activity is higher than older. For the older individuals, antimicrobial activity is higher than the younger individuals. We also looked at specific expression of certain important immune molecules, for example, antimicrobial peptides. And you could see, so these are two such representative AMPs or antimicrobial peptides. You could see that in older individuals, these AMPs are overexpressed, are highly expressed than compared to the younger individuals. And finally, this is an interesting molecule. I would like to keep this molecule in mind. I'll come back to this again. And there's something called phenol oxidative response. This is an insect-specific response. This is an enzymatic response. And this also shows to be, appears to be higher in older individuals. Now the question is, when you see this whole bunch of immune molecules are overexpressed in older individual, what do you expect in terms of fitness benefit? So usually people always connect immune response as something which gives you a benefit in terms of fighting infection or something. So we want to understand that. And the next question that we asked, that does higher immunity help older individuals in surviving better against infections? And we found this. You could see that despite having higher immune components, higher immune responses, older individuals, this is a red line, older individuals are still dying faster than younger individuals. That means the immune response that we just observed is not doing any good to the organism. So organism is still dying faster than the younger individuals. Exactly. So yeah, you're right. So therefore we try to compare, this, you could see this gray line, which is the older individuals without infection. So if difference between this gray line and this red line is higher compared to this green and blue. So we could calculate certain parameters which would tell you that, this ratio of, this between the gray line and red line is higher than the ratio between green and blue line, right? So therefore, I think, of course, now you could see there's a absolute mismatch, right? Having higher immunity, but no fitness benefits. So we thought of, try to understand some sort of mechanism underlying it, right? So while doing some bunch of other experiments, while we're trying to, dissect this organisms out, we observed that once you take off its skin from the dorsal side, you see these black dots all around in older individuals after they've been infected. So later on, we got to know that these are actually melanin deposit because of this phenol oxidative response, right? And in the literature, it has already been shown that this phenol oxidative response is highly cytotoxic for the organisms, right? So by looking at this, so if we compare between the young and older individuals, these black dots are exceedingly higher in older individuals. So we thought of, testing whether this melanin deposits could be an explanatory mechanism, whether this, this black dots or the deposit of melanin could explain the higher mortality in older individuals or not. So we thought of, trying to focus on now the phenol oxidative response in these insects. So what we did is actually we started trying to quantifying the degree of immune damage in these beetles. So this is a method. So you could see this thread-like structure in the beetle here, right? So I don't know how clear it is. So this is called Malpighian tubules. These are equivalent to kidney in these insects. And one distinctive feature of this organ is that these organs, because of their functional requirement, they cannot be covered with a membrane. So they're practically exposed, or they're practically swimming in the let's say the insect blood. Right. So whatever immune reactions, and the insect blood is a place where most of the immune responses happen. And this organ is practically exposed in that hemolymph. So whatever immune response are happening in the blood, they can directly damage this organ. So we want to test that. So what we did, we just dissected out the Malpighian tubule from the beetles, and we measured their fluid secretion rate. As I said, it also acts as insect kidney. So these are one of the major organs which takes part in fluid secretion. So we wanted to measure the fluid secretion ability of this organ, and this is how we measure. So basically, we take a buffer in which we suspend the organ. So this organ has, after dissection, it has one closed end and another is an open end. So we keep the closed end inside the bubble, and the open end we take it out, and this entire thing is immersed in some sort of oil layer. You could see after some time some sort of bubble forming at the open end, and this is what we measure. So, and I think it's very easy to understand that, higher the volume and the bigger the size of the bubble, you would know that the physiological function is better for the particular beetle. So what we found here, so this is what the organ function looked like. So if you compare the young individual, both infection and sham infection, SI is a sham infection, that means it's been injected only with buffer. Whereas infection means a real bacterial infection. You see, even with infection, even in younger individual, there's a sharp decline in their organ function. Compared to that, if you see the older individuals, by virtue of the age, their overall standing fluid secretion ability is already low, but with infection, the organ almost stops functioning. Now you can imagine what happens if a vital organ of the body stops functioning. So which means that the immune response is actually doing really messy in these beetles. So we found in older beetles, kidney almost stops functioning after bacterial infections, and somehow it suggests that there's an increased kidney damage caused by immune response. However, we now wanted to find out the causal link between immune response. So I think so far whatever we're seeing is just some sort of correlation. So we wanted to find a causal connection between this immune response and this organ damage. So what we did, as I said, I think one of our candidate molecule was the phenyloxidase response, because that molecule, that enzymatic pathway is fairly, so they release a lot of free radicals during immune response, and it's possible that free radicals generated by that immune response could be, is responsible behind this sharp decline in their Malpighian tubule activity. So yeah, so I think this is the immune response pathway I was talking about. You could see that, so they basically form melanin deposits, they surround the microbe like this, and they also kill the microbes by generating ROS, they actually generate ROS production here. And you see this particular molecule, which is DHI, it's called dihydroxyindole, which is highly cytotoxic. It's already been shown that this molecule is super cytotoxic. It's really caused kind of serious damage in the insects. So we thought of knocking out this particular immune pathway in our beetles. So you could see that, again, the sham-infected beetles, they were secreting, the secretion rate is higher. Moment you infect them, there's a sharp decline. But then in this particular treatment where we knocked out this particular immune molecule, you see an immediate increase in their, the function of the organ. And subsequently, this actually translated into higher survival. So this is what you see in older individuals. The black line is a sham-infected one. This green line is older beetles, which were infected. And moment we knock out this particular molecule in this, you see a rise in their lifespan. So we were able to rescue the lifespan after knocking out this particular immune response, which somehow suggests a causal connection between that immune response and the survival after infection in older individuals. So therefore, we could summarize certain features of an aging immune system. For instance, it looks like immune response is indeed overactive with aging. There's an increased risk of immune-induced damage to critical organs. And it looks like suppression of overactive immunity is beneficial for older beetles, something which assured us that there's a causal connection between immune response and this damage caused by, there's a causal connection between immune response and the damage. So next, we wanted to understand another very interesting aspect of an aging immune system. So again, coming back to the standard model of, that I tried to tell you in the beginning that, with age, the immune response could be losing its control over, and therefore it becomes now poorly regulated. Now, look at this particular image. Is it very difficult to imagine that with aging, the immune response could be more non-specific? In younger individuals, it is known that in certain organisms, maybe only few molecules or few immune pathways could be enough to control the infection, right? While in case of older individuals, if the regulation is poor, it is possible that immune response becomes more non-specific. Practically, one can fire a cannon to kill a mosquito in our older individuals, and which somehow also goes well with the idea that immune response becomes overactive with aging. So at this point, we stumbled across this very interesting person, this is Bruno, who is also my collaborator, and he published a very interesting paper in Drosophila. So he was working with these antimicrobial peptides, which are host-encoded antibiotics These are short cationic peptides which involve innate immunity. So what he did, he tried to, in his assay, well, his assay was nowhere related to aging of immunity, or he was not interested in any evolutionary aspect, but it is a purely mechanistic paper where he tried to understand, he took a combinatory approach to explore different AMP interactions, right? And what he found very interestingly that in younger flies, so that was his model, that was his experimental, flies. So they were basically young flies, and he could find that these AMPs, so there are a bunch of, there are at least 14 AMPs known in Drosophila, and it looks like that not all AMPs are involved during a given infection. It's only a subset of AMPs which are enough to give you some protection. So, and he showed they actually functions additively or synergistically to provide specific protection against gram-negative bacteria, right? So, to just give you a brief idea about some known insect pathway which are involved in generating AMPs. So there are two known pathways like IMD pathway and toll pathway, and they have their respective set of AMPs. So IMD pathway is usually, they're usually, so this IMD pathway is actually usually expressed when there's infection by gram-negative bacteria, and this toll pathway is usually overexpressed when there is infection by gram-positive bacteria, and both of them produce their independent set of AMPs. So therefore, I think one of the first things that Bruno did is he actually deleted all known AMPs from this Drosophila. So basically, you could create fly without practically no AMP activity. Not only that, he also created a bunch of different combinations of these deletions. You could see in one set of group, for example, if you look at group A, he deleted all AMPs which are toll regulated and which are actually involved in, which are actually involved in resisting gram-positive bacteria. He also deleted a set of, AMPs known as group B, for the time here. For example, it has different molecules, atacin, piperacin, drosomycin, and which are practically regulated by IMD pathway, and they're also involved in fighting gram-negative bacteria, and also create another set which are known as antifungal in nature. He also combined these different groups. Let's say in one set, he deleted both group A and B, and therefore you have this group AB. Another set, he deleted group A and C, so therefore it becomes A and C like this. So it's a basically additive, knockdown of all known AMPs. So I thought of using this particular, set of, resources to understand or, to address my own questions, whether immune response becomes more specific. And there are some multiple experiments that he did. He suggests that, not all AMPs are required to fight infection. Let's say in this case, so this name is not important. Let's say the fly is infected with something, and you see only this AB and BC, which are important. So these are the pink line and this blue line somewhere. The light blue color line somewhere. And these two combinations are most detrimental for the fly. So if you remove them, they can't survive. Practically, and he also noticed this B is common between both the combinations. So he subsequently wanted to understand whether something lies with B only or not. So therefore, he looked at AMPs, which were included in the B combinations. And you see what happens. He found that individual AMPs are not required to kill pathogens. It's basically a combination of AMPs which are often required to kill a pathogen. So you could see that there's an absolute synergism, a lot of, I would say a lot of coordination between different AMPs to fight infections. He also could find some of the cases where he knew that, some infections can be removed only by one AMP. So it is that specific. And this paper is crucial because so far, people who are arguing that insect immunity is non-specific, right? And now with this paper, we know that even with the simple immune system in insects, they could actually elicit a response which is very specific. A traditional feature of, it's a kind of traditional feature of immunity by hard animals, right? So I thought of, using this fly lines in my studies. So what I did, well, again, a very simple question: does aging lead to loss of specificity of immune response? So and then two of these students of mine, they were trying to. So Arun is an exchange student who came to my lab in the summer, and Vishwajit he works with me as a project assistant, and they were trying to look at this problem, and they were trying to do a series of infection, using these different A mutants. So, and it's a very simple experiment. We generated fly group of two different ages, like 3-day versus 25-day old, very simple setup. We took 2 different pathogens, both are gram-negative, Pseudomonas entomophila and Providentia deadgeri, and we just recorded their mortality. What we found is really interesting. So you see in young females, so this is a data on females, in the young females, only AB group, if you delete the AB groups from these flies, they become super susceptible. But in case of older individuals, it seems they need both AB and BC. So there is an additional. So in younger individuals, it needs only AB, but in older individuals, it needs another combinations. So suggesting that the recruitment of more AMPs in older flies. We found somewhat similar result in another bacteria also. Well, somewhat differently. You could see this is a study with Pseudomonas entomophila. And what you see here, both in young male and young female, it seems moment you knock out BC and AB, this starts becoming susceptible. We also tried this combination of the AC, which was not significant. In older individuals, we found the same bunch of AMPs are responsible. So practically, we did not find an evidence that there are more AMPs being recruited than in older flies and younger flies. So both young and old, across age groups, same group of AMPs contribute to post-infection survival. However, when we looked at the single mutants, so something very interesting thing popped up, right? So in case of entomophila infection, it seems that flies that lacked IMD regulators, if you remember the two pathways that I showed you, the two, one is IMD pathway, another Stoll pathway. So only IMD regulated AMPs are important here. Flies which lack IMD regulated AMPs, they are susceptible to infection. So if you follow, even if you don't follow all the colors, I just try to highlight it by making the circle here. So, and it means, and these are the AMPs which are important for both the groups. So you could see that only IMD regulated. However, if you look at the AMPs which are regulated by Toll pathway, they are not susceptible. So it seems that Toll regulated pathways, Toll regulated AMPs are not at all important for younger flies. However, this thing changes completely when you look at the older, if you look at the older flies. What happens in the older flies, you see that, the flies which lacked these IMD regulated AMPs, they are susceptible, as I just shown you in case of younger flies. But even in the flies which lack Toll pathway are now also susceptible. So that means now Toll pathway related AMPs are also playing a role, suggesting now in case of older individual, they need both IMD pathway and Toll pathway to resist infection. And which is something, an indicative of that, with aging, there's something wrong with the specificity of the immune system. So again, this study is still ongoing. We have to cover a lot many more combinations too, but this is just a overview of what we have found so far. And based on this, we try to conclude that maybe the older flies require more AMPs than younger flies to survive after infection. So which is an indicator of non-specificity. Therefore, I think so far we have been able to identify a bunch of characteristics of an aging immune system, which also talks about deregulated state of its state. As I said earlier, we showed that immune response could be in older individuals could be highly cytotoxic. They induce more immunopathology. They seem to be overexpressed, and now we know that they're also non-specific perhaps, right? So overall, this is where our current understanding of my aging, so this is where my understanding of the immunity with aging stands here currently, and now we're trying to understand the next big question. So far what I have been doing, just to trying to scan in some sort of features of an aging immune system, now we have to understand that what actually triggers this transition. Why in older individuals immune response becomes deregulated. So that's what we are now interested in. So in order to do that, again we fall back our age-old hypothesis that we think there has to be some role by the reproductive schedule of the population, right? So so therefore, the simple hypothesis is that if an organism, population is aging faster, right? I think in a way. Let's say if you take a population which are early reproducing, right, you could expect the strength of selection could be declining faster in that population, right? And this could lead to early onset of immune deregulation. Whereas in case of late reproducing individual, the selection stays stronger for a longer time because it has to keep individuals in a better shape for a longer time, right? And which is possibly not the case in early reproducing organisms. And this is what we are doing. We are currently using the this experimental evolution line of flies, where we have two different lines which are fast reproducing and late reproducing. So basically we have fly population which are they actually reproduce just after 8 days since the egg collection. It's pretty fast. And there's another group of flies which are reproducing after 26 days post egg collection. You could see the difference between the aging, difference between the reproductive schedule. And we hypothesize that if you think of rate of increase in immunopathology in these different lines, this is possibly we are going to get. And we think this is something that we are slowly getting because we have data for one block. And I'm not very confident of, but it looks like I think this pattern might follow something like this, that in early aging fly populations, the rate of immunopathology will start increasing faster. We also want to test, so I think if you just take a look at the entire list of different features, I think we are testing all of them. So whether the immune response will become non-specific or not, with and that the time of onset differs between fast reproducing and late reproducing populations. So here, so I think this is where the current this is the current state of the project. We are hopeful to get this thing to be out soon. And but after that, I also got interested into an added perspective to cost of immunity. So now if you look at carefully this picture, it's the same thing that I showed you earlier about phenyl-oxidative response in insects. So many molecules, immune molecules, are cytotoxic in nature, as I said. And if you take an example of this phenyl-oxidative response, and this particular molecule is highly cytotoxic, as I said, and they produce a lot of ROS. So while this could be beneficial to clear of infection, they could also possibly have a damaging consequence. That's something that we have already covered. But what we haven't covered, a new aspect is the following, that, well, as I said, this reactoxin species, they're an important killer of the pathogens. They could also raise the cost of inflammation. They could also damage DNA, which is fairly well known, that immune response, if the inflammation is chronic in nature, it tends to damage DNA. Not only that, it also damages the DNA repair machinery. Now, if you combine the both, you're having DNA damage and your DNA repair machinery is not working well. So it could possibly, well, I think this is something the role has already been ascertained in cancer. But, well, I'm not going to talk about cancer here, but I'm going to talk about another aspect of it. As I said, if both of them are compromised, you could imagine that, so if the DNA repair pathways are going bad, there's a possibility the germline maintenance could also be compromised. And you can imagine if the mutations are going unchecked, in a case of female, or let's say the mutations are going unchecked in an organism which cannot invest a lot in germline maintenance, you could expect there's a rise of mutation transmission to next generations, right? So, and subsequently, if you allow it to build up across generations, it will also lead to the build up of mutation load in a population. So this is a simple, well, I think this is a very oversimplified version of the entire thing, but I just want to test that, whether immune response or immune infection can increase the germline, whether immune response can, they can affect the germline maintenance or not. So in order to do that, I used this, I did this experiment in both two organisms. So one is fruit fly, another is, in flower beetles. The fruit fly experiment has already, fruit flies are already a known model system for this kind of experiment. There are a bunch of experiments on that. So what we did, so this question is this: does infection and immunity reduce germline maintenance? So this is the first question that I want to ask. And we try to estimate by analyzing the fitness effects of dominant mutations transferred to subsequent generations. And in order to do that, what we did, we tried to exploit the following feature of this, the insect system, insect females. So in many insects, including Drosophila, the females, they repair, sperm DNA, the sperm DNA prior to fertilization with the help of the repair proteins in their eggs. And in this system, also males, they cannot repair mutations in their sperm, right? So therefore, one could actually test the ability of the immune challenged females, the ability to fix germline DNA, and therefore you could test with how much it has been transferred to next generations. So this is a brief protocol of what I followed. You can see that, so mutations are being transferred from the male itself. So male has been, they're mutagenized using gamma radiation. So, this is where so the male sperm has been mutagenized using gamma radiations, and they have been mated with two categories of females. One is infected, another is sham infected, both of them receiving mutagenized sperm. We also have a set of controls. And after that, at F1 generation, we relaxed selections. You could see that, you could create from them, you could imagine the progeny would be carrying the half of, sorry about this is a formatting error. So both of male, female should look like this. So half of the genetic material will contain mutations transferred from the male sperm, right? And we measured the mutation load in a F2 generation. So this is basically the number of F2 offspring produced by F1 mating pair descending from the irradiated F0 males relative to number of F2 offspring produced by F1 mating pair descending from respective F0 control males. So you could compare this to value and calculate the effects of mutation transferred. And okay, I think, and then if you could now see the result, it's quite, it was quite, along the line that I predicted here. In both the organism, there's sharp increase in mutation load with infection, right? So although this looks like a standalone data, but this could also have a very important consequences on how evolutionary pathogen resistance would occur in nature. Imagine a population is living under chronic infection. It has to evolve a strong immune response, but immune response is also going to, so immune response is also supposed to induce this kind of cost. So I think there has to be a very sensitive balance between the cost of immunity and benefit of evolving higher immunity. So, but then I think, so currently we're testing this hypothesis again using the experimental evolution, so where we're evolving flies and beetles under chronic infection status, and we are planning to measure their germline maintenance after a certain number of generations. So we expect that it could be possible that to reduce, so as I said, I think certain immune pathways are more cytotoxic. So it's possible that an organism might evolve a strategy where they start using less toxic immune molecules. So there could be a shift in immune strategy from highly toxic immune responses to less toxic immune responses, right? Of course, they could also increase. Well, I don't know about that in particular, but anybody have used this or not as an immunosuppressant or something? Or you said boosting immunity. Well, they're not chemicals, but you could use synthetically designed chemicals, for instance, if you can mimic something of bacterial cell wall components. Let's say peptidoglycan molecule. These are not live, self-replicating molecules, but these are antigen basically. So if you introduce the antigen inside the insect, automatically there will be an immune response. Yeah, I think something. I think another point that I forgot to mention here is actually, so here I'm talking about the effect of infection, but another way to look at the cost of immune activation, where we don't have any infecting pathogens, but just to overactivate the immune response, and that's something we're kind of figuring out. They do have, yes. So this is also they're also kind of feature of an aging immune system. So, and one possible mechanism is I think people have found out is basically because of the leaky gut. So I think the gut microbes, they start getting into the hemolymph, which somehow raises the immunity in older individuals without practically any other invading pathogens. So it's basically your own microbes are now contributing to your immune response. No, so what we're doing, we're introducing mutations through males. So here the females are not mutagenized. So females are only treated by infection or no infection. But the sperm is coming from we have a group of males which has been mutagenized. So there's no difference in male, so males are practically the same, and they're actually receiving the gamma radiation, and their sperm is actually getting mutagenized. So it's the same source of same males, and then it gets into different kind of females infected. I don't think so, because see, if you look at the protocol also, so we actually don't measure the mutation load at generation F1. So this F1, right? So basically this practically takes away the reduce the paternal effects of irradiation treatment. So therefore you can actually now, so here you actually nullify selection on all but unconditioned, you actually all but those mutations which are lethal unconditionally, they will not be here. But I think this, but in this generation you can actually nullify the effects from the father itself. No, we are not looking at the lethal. So that's what. So here basically what we're doing at the F1 generation, we are not taking the lethal mutations, the individuals which has this lethal mutation, because they will not survive. Because what we want is F1 offspring which can reproduce. And therefore we measure the fitness at F2 generations. That's what I'm saying. So yeah, I think there's a bunch of experiment which is still remaining. I think we still need to understand whether after going through several rounds of selection, so, after going through experimental evolution with chronic infection, they could actually increase the germline repeatability or not. And also, it would be really interesting to find how they trade off with other competing biological functions. So this is where I'll stop. I would like to thank, well, I think a bunch of nice collaborators. Bodhi, who is in Isaac Berhampur, who's been, in donating the fly lines with different rate of aging, and Bruno for supplying, Bruno and David, who have been helping me with this mutation load experiments here. And also, this is my lab. Well, I think just want to spread the news that we'll be recruiting PhD students 2020, and so it's a new lab, but I think with a lot of promise, and there's a bunch of very interesting people. Well, I think I'll be happy to take questions.